Okay, sorry about that. I had to check on something. Moving on. So, for question 2, part C, yeah, there are 16 sister chromatids in prophase 1 for this fruit fly. Then, while a fruit fly cell is in metaphase 2, so in metaphase 2, the cell is already split. So there's 2 cells, and each one has 8 chromosomes, and each chromosome is made of 2 sister chromatids. So 4 chromosomes, 2 sister chromatids for each chromosome, which means that there are 8 sister chromatids in metaphase 2. Then part D, a blink is a type of genetic cross. Okay, the answer is test cross. Hey, sorry, I'm making a video. Sorry. So the answer is test cross, where you cross 2 things and see what happens as a result of it. And question part E, a non-functional allele is a null allele. Sometimes it's a recessive allele. A partially functional allele is a hypomorphic allele. Now, next part. Mendel's law of its independent assortment is the first one. It involves 2 traits, because independent assortment is that 2 traits do not affect each other in how they are inherited to the next generation, which is true for the traits that Mendel was testing for, but not for all traits. And the law of segregation only involves 1 trait, because segregation says that if you have 2 homologous chromosomes and there's two genes, one on each chromosome, those 2 alleles will be inherited independently of each other. They will segregate and be inherited independently. So, like, for example, in the case of the purple flowers, the purple was controlled by the dominant allele and white was controlled by the recessive allele. Those traits are inherited independently of each other. They segregate and go separately from each other. Great. Now question 3. Hang on 1 second. Okay. Suppose an organism has the genotype big T, big R, little r, big Y, little y. Assume that the big T, little t alleles, the big R, little r alleles, and the big Y, little y alleles assort independently. And the first part is they want me to diagram something, which I did that already. Based talking about how in metaphase you have the metaphase plate, and you have chromosomes here and their homologous counterpart on the other side, and they want me to illustrate how they are arranged. And so what happens is you could have maternal up here and paternal down here, or you could have some paternal up here and some maternal down here. You could have all kinds of combinations, which they are all illustrated already in my diagram. Indicate all of the possible gamete genotypes that can be produced in this organism at the conclusion of meiosis II. So, since the first 2 traits are big T, means all the gametes will have big T, no matter what. Then some will have big R, some will have little r. So you could have big T, big R, or big T, little r. And some will have big Y, little y. So you could have big T, big R, big Y, big T, big R, little y, big T, little r, big Y, or big T, little r, little y. Okay. How many sister chromatids are found per cell in this organism during metaphase 1? Okay, so in metaphase 1, the cell has not divided yet. It's still one piece. If these genes assort independently, that means they follow the law of independent assortment, which means that each of these genes for height and shape and color has got to be on a different chromosome, which means that there are we know that there's got to be at least 6 chromosomes, 3 for each trait, and the 3 homologous pairs. So how many sister chromatids are found? Okay, so for each chromosome there are 2 sister chromatids, which means that there are 12 sister chromatids at this point. The next question, how many chromosomes are found per cell in this organism during metaphase 2? So after metaphase 1, after just a few steps, the cell divides. So now there are 3 chromosomes in each cell, and for each chromosome, again, there's a sister chromatid, or rather there are 2 sister chromatids for each chromosome, which means that each cell in metaphase 2 has 6 hang on one second. How many chromosomes? Yeah, I'm sorry. There's 3 chromosomes per cell in metaphase 2. Yes. Sorry about that. So, question 4. A horse has 32 pairs of chromosomes. Okay. So, part A. How many chromosomes are present per horse cell at the conclusion of meiosis 1? So, at the conclusion of meiosis 1, what happens is, so there's 32 pairs of chromosomes. So there's 32 chromosomes, and there's 32 more that match up with them, which means there's a total of 64 chromosomes at the beginning. So at the conclusion of meiosis 1, the cell has split, and there's 32 here and 32 here in their own cells, which means there's 32 chromosomes per cell at the end of meiosis 1. And part B, how many tetrads are present per horse cell during prophase 1 of meiosis? So in prophase 1, it's still one cell, which means there's 32 here, 32 here. Now a tetrad is where you have a chromosome and its sister chromatid. Then you have the homologous pair, the 2 chromosomes, and they each have their own sister chromatid. And then the 4 sister chromatids come together to form a tetrad. Okay, sorry, I'm making a video. Huh? I'm making a video. It's okay. So there's 4 sister chromatids for each pair of chromosomes. And since there's 32 pairs of chromosomes at the beginning, it has not split yet in prophase 1. That means there are 32 tetrads. And part D, question 4, part D, how many sister chromatids are present per horse cell during metaphase 2 of meiosis? Wait, I'm sorry. I skipped part C. How many sister chromatids are present in horse cell during prophase 1 of meiosis? Okay, so there's 32 pairs of chromosomes, which means that there's 2 chromosomes per pair, which means that there's 64 chromosomes. And for each chromosome, there are 2 sister chromatids. So 32 times 2 is 64. 64 times 2 is 128. So in prophase 1, when it has not split yet, there's 128 sister chromatids. And part D, how many sister chromatids during metaphase 2? So it's already split. So you just take 128 divided by 2, which is 64. 64 sister chromatids. And in part E, how many bivalents, which is the same as a tetrad, are present per horse cell during anaphase 2 of meiosis. So in anaphase 2, the cell is already split, which means that the two homologous chromosomes and their two sister chromatids have already been separated from each other, which means that there are no bivalents, there are no tetrads. There are no tetrads in anaphase 2, so the answer is 0. Okay, now question 5. Consider the following okay, so there are two pea plants with a certain genetic makeup. Then it tells us what each trait is and what each allele gives us. Okay. Assume that the alleles of all 4 genes assort independently of each other. Okay. So, part A. What proportion of the offspring will be tall, round, green, and terminal? Okay, so tall is a dominant allele. Round is also dominant. Green is recessive, and terminal is also recessive. So I did the same thing I did before, where I yeah, here it is. Pardon me. Where I took all the traits and I crossed them independently on 4 monohybrid crosses. And so, pardon me for one moment. Okay, so for part 5, the monohybrid cross, there are three gametes that have a big T, which means they have the tall allele, and tall is dominant. So 3 of the gametes will have the tall trait, which is dominant. So the zygote that they produce will grow to be a tall pea plant. So that's 3 out of 4. And same thing for round, 3 out of 4. Now for